everyone, welcome to Positron Academy, and today's topic of discussion is on gallium labeled fibroblast activated protein inhibitors, or most popularly known as FAPI, from tumor biology to imaging. And these are the contents for today's presentation. We'll start with understanding the basic tumor biology, followed by we'll study the fibroblast activation protein, or known as the FAP protein, and this protein is central for us to understand the FAPI imaging as a whole. Then we'll talk about some clinical relevance of FAPI. Then we'll talk about radiolabeling and imaging and how these radiolabeling and imaging are being done. Then we'll talk about the battle between FAPI and FTG. So before we go and talk more about FAPI as a whole, we'll spend some time understanding the basic tumor biology, and that's the normal epithelium which progresses to multiple phases to from a pre-malignant dysplasia followed by carcinoma, and what actually happens in each phases. So that's how a normal epithelium looks like, and you can see we have keratinocyte in the epidermis, and we have a stroma, and these group of cells over here and this group of cells over here are separated by a well delineated basement membrane. And in the stroma over here we have collagen bundles, we have fibroblasts, we have monocyte, we have macrophages, we have granulocyte, and so on and so forth. So that's how a normal epithelium looks like. But due to some external factors like mutation, like radiation-induced damage, and multiple other factors, the differentiation of this normal epithelium can be disturbed. And because of this disturbed differentiation, this epithelium can turn out to be hyperplastic epithelium over here. And also, along with changes in the epithelium over here, there are changes in the stroma also over here. We can see increased number of collagen bundles, we have increased number of macrophages, and also we have increased number of capillary tubes over here. And also the fibroblast, which was initially a quiescent fibroblast or resting fibroblast, has now become activated. So in some way we can say there is a kind of crosstalk that is going between this epithelial cell and the stroma, and the basement membrane in this pre-malignant dysplasia, the basement membrane is intact over here. But due to the proliferation of this epithelial cell along with development of these stroma as a whole, these things can turn out to be more havoc. And we can see that the ECM components over here in the carcinoma has become degraded. In this case, we can see the collagen bundles were seen as bundles, but now these are seen as some discrete pieces. And also number of inflammatory cells, macrophages, has increased a lot, and also the stroma has secreted some protein, some enzymes and all, and which has degraded the basement membrane. And this basement membrane and this leaky membrane act as a pathway for these cells to get out of here and go and spread to some distant sites, causing metastasis and all. And also we have a high number of leaky tumor vessels also, and moreover there is activation of the tumor stroma, which lead to the persistent angiogenesis of the lesion. And that's how these carcinoma looks like, and these are the features which are seen in carcinoma. We have a proliferating group of endothelial cells which keep on proliferating. We can see we have blood vessels over here which are now expressing VEGF receptor, and this VEGF receptor is pretty special because it because this VEGF receptor, when binding to the growth factor, it induces angiogenesis. And also we can see some fragments of these ECM molecules like collagen fragments. We can see some increased level of active proteases, and these proteases have break down this basement membrane, and also we can see a broken basement membranes. These are some features which we can see in the epithelium which has progressed to a state of carcinoma. And moreover, one important thing which you can see observe in carcinoma, that the fibroblast initially fibroblast has now transdifferentiated to myofibroblast in carcinoma state. So these are all myofibroblasts which were initially fibroblasts. We can say these are all quiescent fibroblasts which has now turned out to be activated. But when these cells go through and further changes in the stroma and multiple other things, these fibroblasts get transformed to or transdifferentiated to myofibroblast, and this myofibroblast is the characteristics of carcinoma. And that's pretty important as of now, because because for FAPI point of view, this myofibroblast is very important, and we'll talk about this thing later. And that's the crosstalk which keep on and that's the crosstalk between the tumor cells and their stroma. As I've told before, that there are changes in the epithelial cells, and leading to changes in these epithelial cells, there are corresponding changes in the stroma. So definitely there is some sort of crosstalk between these epithelial cells and the stroma, and it has to by means of some enzymes, hormones and all. And the tumor over here, it secretes some growth factor, and this growth factor in turn induce angiogenesis and also recruit some inflammatory cells, fibroblasts, and these things further secrete some proteases, and at the end it lead to the activation of the stroma, and this activated stroma will lead to the tumor malignancy. It promotes malignancy of the tumor, and that's the main reason why you see higher mets. So that's how these things goes on at the tumor biology level. But now the question will be definitely, but how these stories are related to FAPI? So let's go back once again. That's, I have told that the tumor cell, it secretes some growth hormones, some protein, it secretes multiple things, and out of these factors are pretty important, like PDGF or platelet derived growth factor, and TGF beta are the factors which are the growth factors are being released by tumor cell as a whole. And this TGF beta is responsible to transdifferentiate the fibroblasts to myofibroblast, which are the activated smooth muscle reactive fibroblast. And these myofibroblasts are also known as the CAF or cancer associated fibroblasts, and this group of fibroblasts are found mostly in the cancer related tumors and also. And these myofibroblasts, these are identified immunohistochemically based on expression of different markers, and the markers which these myofibroblast expresses, these are alpha smooth muscle actin, vimentin, desmin, and most importantly it expresses this protein called as the fibroblast activation protein. So now we can see that in case of a tumor which has become activated, which has reactive tumor stroma, it actually transdifferentiate fibroblast to myofibroblast, and this myofibroblast is known by its expression of multiple markers, and FAP is one of the markers which the myofibroblast expresses. So now let's spend some time understanding the protein or the enzyme fibroblast activation protein. So what this FAP is all about. So as of now we'll not utter this long name fibroblast activation protein. We'll just simply call as FAP. So this FAP is basically a type 2 integral membrane serine protease of the prolyl oligopeptidase family, and that's the three-dimensional structure of this FAP protein over here, and that's an enzyme named as serine protease. So the specialty of this group of enzyme is that it cleaves the peptide bonds. So that's a peptide over here, and there are multiple amino acids in the peptide. So the main specialty of the serine protease or this FAP protein is that it cleaves right after the proline amino acid bond. For example, that's proline in the peptide, and right after proline we have glycine. So this FAP will come and thus cleave right after proline. So it will just cleave wherever it will find proline. So that's the specialty of this FAP protein. So it cleaves the peptide into multiple small fragments, and where it cleaves the peptide, wherever it finds a proline residue. And this enzyme is known to have some normal roles in normal development process during embryogenesis and tissue remodeling. And apart from these FAP levels are not that much significant in normal adults. We don't have expression of FAP, but there are some examples where we can see expression of FAP. Example in uterus, in cervix, in placenta, in breast, in skin. It shows some moderate expression of FAP compared to tumors, and as a whole, tumors really have higher expression of FAP compared to any other normal body structure, normal body tissue. But this group of like uterus, cervix have they have quite moderate expression of the tumors, and the high expression of FAP is seen in wound healing, in inflammation like arthritis, atherosclerotic plaques, and also in fibrosis we can see higher expression of FAP, and also after myocardial infarction. And moreover, the FAP expression is pretty high in more than 90% of epithelial cancer. The FAP expression is very, very high. And obviously, if FAP is present in our body, it must have some role, and it must have its own substrate. And these are the substrate in which this FAP actually act on. Example, neuropeptide Y, beta-time natriuretic peptide, substance P. And these are examples of the most efficiently hydrolyzed substrates of this FAP enzyme as a whole. Since FAP is an enzyme, so work on FAP also focused on either working on FAP substrates and FAP inhibitors, and our today's topic is more inclined towards understanding the FAP inhibitors. So initially some work has been done to develop FAP substrates, and also some products all have also been developed using FAP substrates, and these substrates were coupled to some cytotoxic drug like melittin, doxorubicin, and so on and so forth, and that's again the three-dimensional structure of the FAP protein. And initially FAP substrates have been developed as a whole, but FAP inhibitors were not that much developed. So in an attempt to develop this FAP specific inhibitor, so this group of people like Cohen, Jason, Lean, Hairbot, and these people, they examined a variety of structurally related molecules, and some of them being very specific to FAP, and these molecules could be used as a lead structure for the new radiopharmaceuticals. And based on their study and several other studies like these, FAPI1 and FAPI2 were being developed, and that's the structure of FAPI1 and that's the structure of FAPI2. And these FAPI inhibitors and these molecules, it showed a great specificity to these FAP enzyme. And apart from this FAP, there are some similar enzymes like FAP, for example, DPP4, CD26, but these FAPI molecules were so specific that they bound to this FAP enzyme as a whole. They do not show any binding to the related proteins like DPP4 and CD26. So these FAPI inhibitors, small molecule inhibitors, are really specific to the FAP enzyme as a whole. So it showed a high specific binding to the human and the murine FAP, and with good and rapid complete internalization. Right, and the FAPI1 was not that much optimal. It was suboptimal because of one factor, is that because of the enzymatic deiodination. We can see that's the iodine, and that's iodine 125 over here, and because once that's inside cell, because some enzymes acts on it, this FAPI1 small molecule inhibitor, and lead to the deiodination of the iodine over here. So if iodine is deiodinated, so obviously we cannot get the signal from the given lesion. So that was not that much optimum, but in contrast, FAPI2 was really excellent compared to FAPI1. And because one of the factor, because FAPI2 was really excellent because it has a chelator of called as DOTA, and it showed better pharmacokinetic and biochemical properties compared to FAPI1. And this FAPI2 was eliminated much slower than FAPI1, but still we'll see later on how FAPI2, how the kinetics of FAPI2, but still FAPI2 is better than FAPI1. But now, because when you started doing the clinical scenarios and all, FAPI2, it showed a great uptake at the initial phase, and up till 1 hour the uptake is the retention of the tracer is very good. But right after 1 hour, retention is very poor. It keep on eliminating the tracer, and because of this reason, keeping FAPI2 as the main structure in mind, multiple other things were being developed. So in order to optimize the uptake and tracer retention of the tumor, so in order to optimize the uptake and tumor retention in the tumor, um, a series of compounds based on FAPI2 was being developed, and FAPI10 was also being developed to enhance to enhance the cellular retention. And what they have done basically, suppose that's the FAPI structure, and they have added some signal for nuclear localization signal, or we call it NLS, and it has results in great intercellular retention and great intercellular transport of this FAPI10. But FAPI10 is not being used as of now, but FAPI4 is very popular in these days. So that's how the things have evolved. We have seen initial development of FAPI1 and FAPI2, and FAPI1 is not that much optimal because of the enzymatic deiodination. This iodine has been knocked out, so it has a pretty high limitation of FAPI1, but FAPI2 is a very pretty good molecule for us to use. But one of the demerits of FAPI2 is that the uptake is very quick, but the clearance is also very quick. So at delayed imaging, we cannot get a good SUV value. So FAPI2 is not that much optimal. And based on FAPI2, a series of compounds were being developed, and like FAPI4, FAPI21, FAPI46, and multiple other compounds were also being developed, which is not being discussed over here. And out of these, FAPI4 is really a potent radiopharmaceutical or potent small molecule inhibitor for these FAP enzyme. So, and we have, because it has a DOTA over here, it's a chelator over here, so we can use multiple radio metals for chelating this chelator. We have used gallium-68, yttrium-90, and lutetium, though used, but it's in clinical use so far. And so we have already explained that the fact that FAPI2 has FAPI2 tumor uptake is decreased by 75% from 1 hour to 3 hour, but in contrast, FAPI4 has this prolonged tumor retention compared to FAPI2. And these are the structure for FAPI2 and FAPI4. And if I want to use this FAPI for diagnostic purpose, we have the gallium ions, so gallium metal, gallium-68, radioactive 1. So we can use these for chelating to this DOTA, and it will make a coordinate covalent bond and can be used for imaging part of view, because gallium, it decays by positron emission, and finally will annihilate to some electron, and gamma ray photon will be released and for imaging. And that's similarly for FAPI4. We can see the structure is pretty similar, except for these two where it's fluorine over here. So otherwise structure is pretty similar. And this FAPI4, it shows a higher retention as compared to FAPI1. It has a higher washout of 75%, but it has a good retention. And within from 1 hour to 3 hour, only about 25 washout is there. But as compared to FAPI2, the washout is pretty higher, about 75% washout is there in FAPI2. And yes, you can see the graph over here. So that's the graph for FAPI, that's the images for gallium FAPI2 and gallium FAPI4. And we can see FAPI2 shows a very quick uptake with higher SUV compared to that of FAPI4. But if you see at later time points, at higher incubation time, it has a quick washout from all the lesional sites. So if I want to take image at later time points, I might not get the optimal SUV value, which will help me to confidently say that's a lesion or not. So that's why FAPI4 is pretty optimal, because it shows a good tracer uptake also, and tracer retention at the metastatic site. And that's the SNMI image of the year. So that's the Nobel radio tracer, which detects 28 cancer types and paving the way for development of new therapy. So with this FAPI, we can image a variety of cancers, starting from sarcoma, starting from esophageal cancer, breast cancer, lung cancer, colorectal cancer, pancreatic cancer, ovarian cancer, prostate cancer. So it helps you to image a wide variety of cancer as a whole. And that's pretty confident for you, because you know that FAPI of this FAP expression is not being done, it's not being expressed by any normal cell. Only those cells which have become carcinogenic, which have activated fibroblast, will express these FAP enzyme. So this image is very confident, is very popular for you to say that's the lesion, and it's give you more confidence compared to other modality like FDG, because FDG uptake is very non-specific. The lesion will take glucose because of its metabolic demand. It needs more glucose for its survival. Apart from this, inflammation, infection also will take up glucose. So there is some some you are not eventually sure about the lesional uptake. It's a lesion or it's a inflammation. So let's try CODI, inflammation, and also it gives you more confidence in your interpretation and reporting. And for example, that's a patient who had colorectal carcinoma, and we can see some lesion in the lungs and some hypodense lesion in the liver. And corresponding to FAPI image, you can see some FAPI avid uptick over here, which actually gives you the confidence to say these are all metastases from the primary colorectal carcinoma. And that's the patient who had hepatitis B with elevated CA 199 levels. And based on the ultrasound finding, the patient has expanded intrahepatic bile duct. But when the patient went for FDG PET CT scan, we cannot see any uptake in this region. But gallium DOTA FAPI4 has shown an uptick at these uptick alongside the expanded intrahepatic bile, and that's the primary side of the malignancy. So, and a biopsy revealed this intrahepatic cholangiocarcinoma, and the mass was successfully dissected. And these FAPI can also being used to improve the GTV, or the gross tumor volume delineation, in the preparation of the patient to EBRT, or external beam radiation therapy. And these are the comparison of the patient who had both FDG and gallium FAPI, and we can see that the patients have similar scans over here. About 5 of 6 patients present similar strong uptake with both FDG and gallium. And for example, in this patient over here, this esophageal cancer, so FAPI has a good uptake at the cancer at the tumor site. And also we can see that it gives you more confidence because of the corresponding mucosa over here. Mucosa has a great uptake over here, but FAPI, there is uptake by the tumor lesion only, no uptake by the mucosa. That's give you more confidence about the scan. And that's the comparative study of FDG and gallium FAPI. And now we are moving to the next part of radiolabeling and imaging. So how do you radiolabel this FAPI? So that's your peptide, or the small molecule which you will procure from some companies and all. And that's how it looks like, FAPI4, and that's pretty good for tumor imaging. And you have to use a radiometal to actually radiolabel this molecule. So what you will do, basically, you have your germanium gallium generator, and you will use some element like 0.05 molar HCl. You get some gallate ions, and then you will mix this gallate to this reactor, to the reaction vial, and this peptide to the reaction vial, give some optimal pH temperature conditions for for about 10 minutes, and finally you will get your gallium right at these chelator. And these molecule will be specific for your fibroblast activation protein, and these DOTA along with this radiometal will give you the signal that that's here I am present. So this gallium will decay by positron emission, and this positron will move around, lose energy, and finally emit and finally annihilate with an electron and release 2 high energy gamma ray photon, and that will be used for imaging. And that's how you get the imaging post the detection of the number of photons along the multiple LO wires, and then reconstructing the image back to it. So that's how this imaging looks like of this FAPI. So now we are entering the last phase of this presentation, that's the battle between FAPI and FDG. So why do the FAPI based tracer, it appeared so promising compared to FDG, mainly for oncological setting? So first is that this DOTA FAPI is labeled with gallium 68, and that's obtained from a generator. And these radio tracer can be produced on site in the departmental in the radiopharmacy lab without the need of an on-site cyclotron, and it makes it more easier at lower cost, even in some small center. And apart from these That's because for every elution we don't get higher activity. So if we have a higher patient load, then that's not feasible enough for with the generator. So we have to think about something else. So so there have been some increasing interest in the production of gallium 68 using medical cyclotron. So what they have done, basically they have a zinc target, zinc 68, and in the cyclotron they're bombarding it with a beam of protons, and finally you end up getting gallium 68 by the emission of a high energy neutron, and these gallium 68 will be used for radiolabeling these particular FAPI4, and finally you will get your gallium 68 DOTA FAPI, and that's being used for the expression for the tumor imaging, and this will bind to those tumor which are expressing FAP. Okay, first point we have talked about it. Secondly, that don't need to go for any dietary preparation. For example, for FDG scans, we have to make patient prepare for the scan. If the patient is hyperglycemic, then it's not optimal for the patient to go for the scan. So we have to go for dietary restrictions. Are there? There are multiple things are there, but for FAPI scans, we don't need to think about those things, because that's not FAPI is not related to any metabolic pathways, which glucose is related to some metabolic pathways. FAPI is no way related to metabolic pathway, so leading to a high patient compliance. And apart from this FAPI is very feasible for diabetic patient, because we don't need to control the high serum glucose levels, which we need to control for the FDG scans. And thirdly, the patient uptake time is reduced to 10 minutes, and that's certainly an advantage for the ill patients, and most probably for all patients that's probably an uptake probably an advantage. And because of shorter waiting time, which can be relevant in sick patient as well as for the departmental workflow, and the radiation burden of the examinations might be reduced if the injector activity can be lessened due to faster uptake time. And finally, if you see the uptake of gallium FAPI and FDG, we can see a lower uptake in brain, liver, oral mucosa uptake. So if there are primary lesion in the brain, liver, oral mucosa, that we can this FAPI can be used. This lesion can be identified more easily by FAPI compared to FDG. So now that's a comparative study which this group of people have done, and that's a comparative study between gallium DOTA FAPI and FDG PET-CT, and that's the study protocol which they have used. I'm not going to detail about the study protocol. Let me talk more about the result part. So for primary tumor detection, these people have found that the sensitivity for FDG PET-CT was about 82.1%. Sensitivity for PET is about 82.1% for detection of primary tumors, and for FAPI, the sensitivity for primary tumor detection was about 98.2%. So we can clearly see that FAPI has a higher sensitivity for detecting the primary tumors as compared to that of the FDG PET-CT scans. And the primary lesions are well visualized with clear tumor delineation, and it demonstrated a higher tumor to background ratio contrast than that of FDG PET-CT scans. And the SUV in primary tumors were significantly higher compared to FDG. As we can see from here, there's a different cancer. For example, for lung cancer with a median size of about 2.7 centimeter, the FDG SUV median value was about 4.5. And SUV median for gallium DOTA FAPI was 11.9, and for every cancer we will see that the uptake, the SUV value for DOTA for FAPI is pretty higher than compared to the FDG of the FDG. So we can clearly say the SUV or the centralized uptake value for the FAPI is higher than compared to FDG. So FDG has a lower SUV, but as that FAPI uptake is pretty high compared to that of FDG. And that's the diagnostic performance of FAPI and FDG. For lymph node assessment, we can see clearly that the DOTA FAPI has higher sensitivity for the assessment of lymph node involvement compared to FDG. So DOTA FAPI has about 86.4% sensitivity, and FDG has about 45.5% sensitivity. But contrastly, the specificity of FAPI is pretty less compared to FDG. So specificity is about 58.8 percentage, but FDG has higher specificity than FAPI, about 76.5 percentage specificity FDG has, and that's for assessment of bone and visceral mats. We can see that again DOTA FAPI has higher sensitivity compared to that of FDG, but whereas the specificity for the bone and visceral mats is pretty low for DOTA, but FDG has higher specificity than sensitivity. So for the depiction of distant mats, gallium FAPI, it outperformed better than FDG, especially in liver, peritoneum, mesentery, omentum, brain, and axial skeleton, confirming the abdominal data that gallium FAPI was far better than FDG in some organs. But because of the limited availability of the tracer and a small number of research done so far, so it requires more evaluation, mainly in different contexts, in order to obtain a significant panel of advantages and hopefully some scarce pitfall for FAPI. So what should it choose? The promising fisherman or the outrageously mature? So time will tell as of now. At least for now, FDG can sleep peacefully. So that's for that's for all today. So thank you so much for watching this video. And if you like this video, please do like, share, and subscribe. And please provide your feedback.